Welcome to Episode 4 of Epigenetics Explained. Today I will be talking about another important epigenetic mechanism, histone modifications. This is not just one mechanism, but a group of mechanisms that target histone proteins, which include acetylation, which is the addition of an acetyl group, methylation, which is the addition of a methyl group, phosphorylation, which is the addition of a phosphate group, ubiquitination, which is the addition of a ubiquitin protein. You get the point. Today I will be focusing on the first two, histone acetylation and methylation. But before I talk about them, I need to outline some background information. There are three main types of enzymes which facilitate histone modifications. Writers, which add marks to histones. Erasers, which remove marks from histones. And readers, which bind to and interpret marks on histones. Histone proteins consist of two parts. A C-terminal domain, which accounts for around 75% of the histone's mass, and an N-terminal tail, which accounts for the remaining 25% of the histone's mass. Histone modifications usually target N-terminal tails because they are relatively accessible to writer enzymes. In addition, most histone modifications take place on H3 and H4, and sometimes on H2A and H2B. There are two main mechanisms by which histone modifications affect chromatin structure and thus gene expression. The first is by introducing a difference in charge. For example, a histone modification could result in a positively charged histone becoming neutral. And second, by recruiting reader machinery. For example, the transcription complex and remodeling enzymes, to rearrange chromatin through various chemical reactions. Histone modifications, like other epigenetic mechanisms, are responsible for transcriptional activation or inactivation. These modifications also play a role in chromosome packaging. This is required in cell division when the DNA needs to be replicated. The combination of different modifications on a nucleosome constitutes the histone code. Although it is relatively easy to figure out the function of individual modifications, the goal for scientists is to be able to interpret outcomes of complex combinations of histone modifications. Now, on to histone acetylation and methylation. Acetylation usually results in activation and repair, though this isn't always the case for the H4 protein. The reason for this is that the N-terminal tails of H3 and H4 are positively charged in their deacetylated form. This allows them to wrap tightly around the negatively charged DNA and form heterochromatin. However, during acetylation, an enzyme called histone acetyltransferase, or HAT for short, adds an acetyl group to the lysine or arginine residues of an N-terminal tail. This removes the positive charge of the lysine/arginine residues and thus loosens the bond between histones and DNA. In other words, the chromatin becomes euchromatin. This configuration increases accessibility of transcription factors to DNA, thus activating the target genes. On the other hand, an enzyme called histone deacetylase, or HDAC for short, removes acetyl groups from lysine residues. This restores the positive charge of lysine residues, thus tightening the bond between histones and DNA. In other words, the chromatin becomes heterochromatin once more. This decreases the accessibility of transcription factors to DNA, thus inactivating the target genes. There are two types of HATs and HDACs. There's the nuclear A type, which targets histones, and the cytoplasmic B type, which targets non-histone proteins. Even though cytoplasmic HATs and HDACs don't directly target gene expression, they can modify transcription factors, which do affect gene expression. Histone methylation can activate or repress gene transcription and expression. This depends on the location of the amino acid residues that were modified and the number of methyl groups added. What do I mean by this? Amino acids can be monomethylated, that's one methyl group, dimethylated, that's two methyl groups, or trimethylated, which is three methyl groups. The addition of a methyl group to the lysine or arginine residues of an N-terminal tail is mediated by methyltransferases, or HMT for short, while its removal is mediated by histone demethylases, or HDM for short. Well, that concludes this episode of Epigenetics Explained. Thank you for watching, and stay tuned for the next episode to learn about one other epigenetic mechanism I haven't mentioned yet, which is non-coding RNAs.